Okay, we're back talking about translation, and once again I'm just going to draw my messenger RNA strand with its 5' and its 3' end. I'm going to have a start codon and I'm going to have a stop codon. I want to go back to talk about the transfer RNAs, and specifically the first thing I want to talk about is charging a transfer RNA, and that what I mean by that is adding an amino acid. So this is done by a particular kind of enzyme. These are quite ubiquitous enzymes. You find them in all kinds of cellular life, prokaryotes, eukaryotes, us humans have them. These are called tRNA synthases, also called tRNA synthetases. And what these do is they carry out the charging of a transfer RNA in two ways. In other words, adding the amino acid. First of all, the enzyme picks up the amino acids. So that's enzyme plus amino acid. Plus, it needs a little energy, so it gets this from the cell, ATP. What that leads is an enzyme plus amino acid complex. So these form a kind of complex. So the enzyme takes it up, takes a little energy out, so it releases pyrophosphate, inorganic pyrophosphate. Two of the phosphates are broken off of this ATP, and so you've got the enzyme and the amino acid complex. Then the enzyme picks up the transfer RNA. So now you've got all of these three together in a complex. And finally, the enzyme is released, and it also releases the tRNA, which I'm going to draw in its cloverleaf structure. Plus, the amino acid is attached to it. Okay? And so this is what's now called a charged tRNA. Now, if we look at how this works on a messenger RNA, I'm just going to draw a messenger RNA out here. And I'm just going to put two sample codons onto it. I'm just going to draw them here. And I'm going to say this codon is the codon A, G, U. And I'm just going to leave a little space just so that we can see it more clearly. I'm going to pretend there's another codon here, and I'm going to say that's the U, A, C codon. Now, it's very easy to look up the genetic code on the internet. You can see it in all of your textbooks. But you can quite clearly see that AGU is a codon that encodes the amino acid serine. So the only charged tRNA that's of any relevance to this codon is a charged tRNA that has a relevant anticodon that can recognize this codon. And the anticodon that could recognize it, and again I'm going to draw another transfer RNA here. Okay. There's a variable loop, the pseudouridine loop, and the acceptor is one that has serine and that has perhaps an anticodon of U, C, A. The three nucleotides that are at this particular location on the transfer RNA must correspond to the three nucleotides of the codon, or else there won't be a fit. This is the classic codon-codon interaction. And the next transfer RNA that's of interest here is a transfer RNA that will have tyrosine. And you can look up your genetic code, you can look it up on the internet, and you can see that tyrosine is encoded by the codon UAC, and a relevant transfer RNA for this will be a transfer RNA that at its anticodon, it has an A, U, and a G anticodon. And this particular codon-anticodon interaction is important for recognizing which amino acid is going to be added to the growing protein chain. So now that we've gotten all the players, I'm going to talk to you about the process of initiating translation. So I'm going to draw our messenger RNA again, like I did earlier on. And what we have here is our start codon, which is always A, AUG. Well, it's almost always AUG. We're going to ignore that for the moment. And then we've got a stop codon here. Okay. What you have in the 5' untranslated region is an RBS. That is to say a ribosome binding site. And the ribosome binding site is what the ribosome recognizes. And I'm going to say that specifically, and I said this in the earlier video, there are two subunits to the ribosome, the small subunit and the large subunit. So we call this the SSU, and we call this the LSU. Small subunit, large subunit. The large subunit has got two cavities. Okay, we're going to call these the A site and the P site. Okay, so this is the P site and this is the site. Here's the A site, and the transfer RNAs will nestle in here as we carry out protein synthesis. But I'll talk about that in a little while. Okay, but the small subunit of the ribosome is the first thing that attaches. So the small subunit attaches here, and what it recognizes is it recognizes the ribosome binding site, and what actually recognizes the ribosome binding site is the ribosomal RNA molecule that I already spoke about that's in the small subunit. And so the small subunit binds on here, and the very first transfer RNA binds on here, and the very first transfer RNA is the transfer RNA that recognizes AUG, the start codon, it has a methionine amino acid, and its anticodon will be UAC. Okay. Once the small subunit has recognized the ribosome binding site and the transfer RNA is in place, the small subunit moves down so that it's positioned nicely over the start codon, and only at this point, the very last thing to happen is the large subunit of the ribosome is joined on. And this first transfer RNA is nicely nestled in the P site. The A site is free. So I'll draw this a little bit more clearly down here just to reproduce this. First codon, small subunit of the ribosome bind, large subunit of the ribosome is bound. The first transfer RNA, I'm going to draw it fairly roughly here. First transfer RNA is bound to the first codon, and the second cavity, the A site, is still available. And there's three codons, or there's three nucleotides, a single codon available here. What's going to happen? The next transfer RNA is going to reside in here. This transfer RNA is carrying with it an amino acid. There's already an amino acid here. It's generally the methionine amino acid. And so the second transfer RNA will come into the P site. I'm just going to draw it over here just to show it a little bit more clearly. So small subunit, large subunit, two cavities, and now two tRNAs present. Two transfer RNAs are present, two amino acids are present. And what happens is that the first amino acid is transferred across to the second amino acid. It's what we call translocated. Okay. The next thing that happens is that the methionine transfer RNA is kicked out. The ribosome shifts up by 3 base pairs, so that now, once again, we have a free P site. Sorry, we have a free A site, and the P site is occupied. It's occupied by a transfer RNA, which is the second transfer RNA, and this has 2 amino acids attached to it. In will come the third transfer RNA. With its amino acid, codon-anticodon rules will apply, and you'll add a third amino acid to the, or you'll add the growing peptide chain onto the third amino acid. This process continues all the way until you get to a stop codon. There's no transfer RNA for a stop codon. And so as a consequence, a new transfer RNA is not added. Instead, a release factor is added. And the peptide is transferred to water and the whole complex is dissociated. And that is the process of amino acid chain elongation.